Hello, everyone. My name is Clancy McIntyre. I'm a senior biology major at Duquesne University, and today I'll be discussing with you all my poster, Characterizing Transglutaminase Activity of the semenogelin-1 protein of humans and chimpanzees. As far as primates are concerned, reproductive proteins are among the most rapidly evolving genes. Reproductive proteins are the most rapidly evolving proteins within the primates. Especially in primates with high sperm competition, among which is the chimpanzee, which in comparison to humans, which are mostly monogamous, are polygynandrous. So many males will mate with many females across both of their lifespans. And again, this leads to high sperm competition. So the competition will occur between males where many males are trying to sire the same female. And it won't be seen in a species like a gorilla, which is polygynous, where one male will mate with many females, where it'll select for, very large body size for sexual dimorphism. You won't see these in chimpanzees. You'll see high rates of sperm competition where each male is competing molecularly between their sperm and their semen. Among these rapidly evolving proteins is the semenogelin-1 protein, which is a structural protein found in the seminal plasma of primates, and it makes up a majority of the protein found in seminal plasma. And as you'd imagine, these rapidly evolving proteins would have enzymes that change along with them over time, among which is the transglutaminase. Which is responsible for coagulation of these structural semenogelin-1 proteins. And what will happen is that glutamine and lysine residues of these proteins will cross-link to create these large complexes of these proteins that will lead to coagulation. And you'll see higher levels of coagulation in these high sperm competition species like the chimpanzee. And what they'll do is form something called a copulatory plug, which is thought to increase sperm motility, as well as physically make it more difficult for other males to sire the same female after mating. And an interesting case of this rapidly evolving protein is between the human and the chimpanzee. Again, human is mostly monogamous, so lower sperm competition, versus the chimpanzee, who's polygynandrous. As you can see, they look pretty similar, except for the 1A fragment of the human that is duplicated 6 times over in the chimpanzee. And I'm under the impression that the coagulation differences between human and the higher sperm competition species like the chimpanzee in this case is because these 1A fragments are actually extra substrates for the transglutaminase to cross-link and lead to higher coagulation. I'll be testing this hypothesis by expressing and purifying semenogelin fragments of the human of each of these fragments. Again, that's 3A through 3B. And introducing them into a gel-based transglutaminase assay to visually see if there's any cross-linking happening in these fragments. Moving down here to figure 1, this is a quick look at my purification process. Again, I'm using a E. coli vector or E. coli system for expression. And what I'll end up getting from my vector is what I call a fusion protein. And what that is a maltose binding protein and histidine tag connected to my semenogelin-1 fragment of interest. And what this maltose binding protein his tag does is gives me 2 different points that I could possibly use for purification, as well as helps stabilize my protein, which in my case, I use an amylase resin to bind and then elute using the maltose binding protein, where I'll get my pure fusion protein. As far as my assays are concerned, I'll be imaging these using UV light and an analog of the lysine residue called MDC. Now, MDC fluoresces under UV light, but it fluoresces even more when crossing with glutamine. So if I do these assays, run them on a gel, I'll be able to see if there's a band of an appropriate size, let's say, of the fusion protein that I'm looking for. I'll know that there's cross-linking happening because the glutamines of my fragments will be cross-linking with the lysine analog MDC. Here's a quick look at my amylase purification. Again, these elutions, these large bands are actually of the same size of my fusion protein. And that's what I'll be using for the subsequent assays. Figure 4 and 5 are the assays that I ran under UV, as well as a Kumasi stain, which I really use more as a normalization to make sure that any fluorescence I see here, I can compare to the general concentration of protein over here. But what I will show you is that all bands, 3A, all the A's up here and all the B's down here, have a band at the size roughly of the fusion protein here that's fluorescent. Again, that's the approximate size of the semenogelin-1 fusion protein, as well as in the 2A. In the 2B, I have it highlighted down here. There's bands at about double the size of the fusion protein I've produced, and another one at about 3 times the size, 4 times the size, and so on up until the well, the gel. And I will cover the what this really means at the end of my little presentation. Just to go back, this MBP column is the MBP, again, that's attached to the semenogelin protein. We run that on a gel to make sure that there's no transglutaminase activity happening, so we can make sure that it's not changing any possible results that we see in these gels, as well as a casein positive control, which is a known substrate for transglutaminase. And again, it just lets us know that every reagent that we put in these assays is working properly. And moving on to the discussion, this figure here is an updated map of the two proteins. And all these check marks, all of them mean that there was transglutaminase activity on all of these fragments. The green meaning that is at least one band here. We know there's a band at the approximate size of the fusion protein, whereas the yellow, the gold check marks relates to not only this fragment having a band here, but at a band at double, triple, quadruple the size. So what does that mean? That means that not only are the substrates, not only are these protein fragments substrates for the glutamine side, because again, we're using a lysine analog to image, but also lysine. So not only are these fragments binding to the lysine analog from a glutamine substrate, but they're also binding to lysines of the same proteins in that solution. So we can then infer that not only are they responsible for a glutamine, but also a lysine substrate on those protein fragments. So what we can consider from these results is that there is at least transglutaminase activity on the 1A fragment. So we can then infer that the extra duplications on the chimpanzee fragment are for extra transglutaminase substrates and for more coagulation in general. Future directions: in the future, we would rather not infer the activity of the chimpanzee fragments, but instead put these into the assays ourselves. And sooner, we would also like to use TGM4. In these assays, we use a TGM2, which is actually a liver transglutaminase, not that of the TGM4, the prostate gland, which again, TGM4 would be more ideal since that's the enzyme found in vivo, but the TGM2 is more widely available, and it gives us a good idea of the general activity of the transglutaminase on these substrates. Here are my references, along with my acknowledgements for my PI, Dr. Michael Jensen Seaman, as well as the Bayer School, all undergraduate students within my lab, and our NIH grant. Thank you very much.